Hello everyone, and welcome to this beginner's video where we will discuss the human cell. And in this video we kept it about 10 slides just to keep it concise, but we will also have an additional 2 sets of quizzes towards the end. So before we get started, let's discuss what a cell is. So the cell theory tells us that for one, all living things are composed or made of cells. This just means that if there is any organism that is alive or living, the theory states that that organism is made up of cells. Cells also are the basic functional unit of life. So without life, we would not have cells. Cells are the building block of life. And also, cells arise only from pre-existing cells. So we can see this part of the theory being expressed with what we call cell division. So a baby is able to become an adult due to cell division. When you have a scratch on your skin and that scratch heals, that is because of cell division. So cells are able to divide from cells that are other cells that are already alive. So just to kind of have an overview of how an organism is made, first it starts with chemicals. You have enough chemicals to create a cell, and then that cell becomes a tissue if you have enough of those, and then enough tissues becomes an organ, and then up to an organ system, and then you can have an organism from that. And this is all because of what we're discussing today, a cell. So it's important to know that we have two different categories of cells. That is eukaryotic and prokaryotic. Now, I know these words are kind of big, kind of different, but we'll break these down and what these are and what they mean. So eukaryotic just means that the cell that is eukaryotic has a true nucleus, and then it also has membrane-bound organelles. Just to keep it as simple as possible, those are the two main things that make a eukaryotic cell eukaryotic. So there are 4 different types of eukaryotic cells. The first one here in the top right, the blue circular looking cell, this is an animal cell. Now these are the cells that we have. Human cells are categorized under animal cells, and there's a reason for that. Always remember that structure equals function. However, a cell in this case, or however anything like a chemical or anything molecule is designed, it's designed for its function. So if you look next to that one, the green rectangular cell is a plant cell. Notice how these two cells differentiate, and there's a reason for that, because plant cells, obviously plants don't look like us, and they also don't typically move like we do. So their structure is going to be different than ours. They have this thick cell wall, that darker green around, and we do not have a cell wall. And that is for the purpose of their function. And if we go further, we have the fungal cell in the right, and then the protist in the bottom. And notice that these aren't as heavily complex with membrane-bound organelles, but they do still classify as eukaryotic. So notice that in each of these diagrams, there is a circular, there's circular organelle, and within that organelle, some of these you can see the nucleolus. But the circular organelle is the nucleus. So you see the nucleus, and then all of the other pretty colorful stuff around that is the membrane-bound organelles. Okay, so if we break down what prokaryotic is, basically it means it does not have the membrane-bound organelles or nucleus. But bacteria do have DNA. So it's important to understand that as we go along and learn what a nucleus is. But it's important to know that bacteria, though we're not going to focus on that today, does contain DNA, but it's not contained in a true nucleus like ours would be. Okay? So here are the main components of a cell. We have nucleus, nucleolus, nuclear envelope. Ribosomes, rough ER, smooth ER, mitochondria, lysosomes, Golgi apparatus, peroxisomes, microfilaments, microtubules, centrioles, centrosomes, intermediate filaments, cytoskeleton, and cytosol. So these are the main components we're going to study today and go over today. And we're typically going to use this picture and this diagram of this cell structure throughout the cell just to keep it consistent. Okay, here is a slide that you can pause, but for the sake of time, I'm going to go ahead and move on. But just to kind of give a realistic kind of representation of kind of what a cell does and the different functions of each organelle inside the cell, I kind of made a parallel to like a city or a town, how a cell can relate to a city or a town and the different functions of that. Okay, so moving ahead, the function of each organelle. First, let's start with the nucleus. So the nucleus, if you see in the top right corner, using that same image, we're going to pull that nucleus out and look at it up close. If we cut that nucleus open, we can see in the inner contents, and what is inside of that nucleus is DNA. So the stringy purple stuff, this is DNA. And DNA can become so condensed and compacted into what we call chromosomes. So if you hear chromosomes, that is because it is DNA being wound up and packed into what we call chromosomes. And there's a reason for that, is to keep things organized so that when cell division happens, we don't have any errors. So mRNA is also made here, where DNA is found, and the reason and purpose for that is to transcribe DNA, because DNA cannot leave the nucleus. So it's kind of like with our brains, okay? So our brains contain very important information to help function the rest of our bodies. Well, our brains are encased in a skull. So it's important that what is inside of the nucleus does not get damaged. Okay, so mRNA is going to assist with transcribing the coded information that we need in order to produce what we need outside of the nucleus. And we'll go into more detail with that. Nuclear envelope, like I said, with the human brain, has a skull. The nuclear envelope protects the nucleus. So it is a protective layer and it surrounds the nucleus, but it has pores and openings, and the reason for that is for the genetic materials that is being carried on a strand like mRNA from the coded DNA sequence is able to leave the nucleus, because in order for us to code for the DNA, because that's all DNA really is just strands of coding. And in order for us to code for what that is, we have to have that information leave the nucleus, and it will leave as mRNA. And eventually code for proteins. Then we have the nucleolus, and the nucleolus's main function is to assist with rRNA. So we have three different types of RNA. We have mRNA, rRNA, and tRNA. And the rRNA is responsible for making this next organelle that we're going to cover, and that is the ribosome. So the ribosomes here in this picture are the orange studs that are connected to the blue around, covering all around the nucleus, and it is studded and gives that rough feature. So ribosomes are proteins themselves, but they also help to make other proteins from the mRNA strand that leaves the nucleus. And here is the process that we see: DNA to RNA to proteins. So the goal from coding DNA is to make a protein, and proteins are going to be made outside of that nucleus. So the rough ER is actually connected to the nucleus or the nuclear envelope, and there is a reason for that, because immediately when mRNA enters into the cytoplasm outside of the nucleus, it is going to be translated to a protein. Okay, so we want that, and once it's translated to a protein, it's going to go further through the next process. Okay, so the smooth ER is specific to producing or making lipids, and eventually we could discuss that more, but that is essentially fats. A macromolecule is called that makes fats, things like that, or lipids. So this makes lipids, but it also detoxifies the cell. And now we have the Golgi apparatus. So remember what I said about those proteins. Once those proteins are being made, it's going to move on to the Golgi apparatus, which is this green area. So notice what the Golgi apparatus does. It packages and releases those proteins. Once they're made, it's going to release those proteins in vesicles that'll kind of pinch off that Golgi, and it can transfer throughout the cell or go to the cell surface, and assist with the plasma membrane. And so that is a really cool organelle to me. And the mitochondria is very important because it is the power plant of the cell. It helps run the cell. It is the energy, the currency to the energy of the cell. So without mitochondria, we would not have ATP, which is that molecule, and it would essentially kill the cell if our mitochondria are damaged in any way. Lysosomes and peroxisomes both assist in breaking down things. Lysosomes help to break down waste in the cell, kind of like waste management or trash, um, um, or dump truck. And peroxisomes help with breaking down things like alcohols and fats, but the main thing is it helps break down hydrogen peroxide into safer components such as water and oxygen. And here we have microfilaments. So these filaments help with the structure of the cell, and they also help with organizing the cell appropriately. So here we have the microfilaments, which protect the cell, assisted in its structure, and a specific protein that is localized with microfilaments is called actin. And so this helps move things around in the cell that need to be moved around. And then we have intermediate filaments. So these filaments help with structure and support and shape, and also elasticity of the cell, because these cells, they're not hard and rigid like that plant cell is. But this cell is able to fluctuate and move, but also have stability and structure. And then we have microtubules. So microtubules are very important because they assist with creating pathways for other motor proteins to move down and move across the cell. They also play a very vital role in, remember those chromosomes we talked about? When cell division happens, these microtubules extend from the centrioles, which we'll talk about next, in order to help organize those chromosomes so they can split properly during cell division. So one of the last slides we have before our quiz is the function of each organelle regarding the centriole, centrosome, cytoskeleton, and cytoplasm, and then the outer border known as the plasma membrane. So those centrioles I discussed here in this bottom box, we have the centrioles, and the centrosomes is just the overall aspect, and the centrioles, which are also made of microtubules or where they're located, help to extend those microtubules out, and it's almost kind of like fingers being extended out all throughout the cell to create and help organize those pathways. And then we have the cytoskeleton, which is the structure of the cell, and the cytoplasm, which is the body of that cell, and contains cytosol, which is the fluid that helps keep things moving easily inside the cell. Then we have the outer layer known as the plasma membrane. It is selective, so it helps protect what is able to enter and exit the cell. So here is a quiz that we can cover. So this is label the diagram, and I'm going to allow you to pause this video because what's next is the actual answer. So I'll let you pause it now. Here are the answers. And last but not least, the critical thinking questions. So you can go ahead and pause, and here are the answers. I hope you enjoyed this video. Thank you. Bye-bye.